Okay, so good morning, everyone. Today we will be discussing something that is like an engine room and gene room of life, and that is the DNA replication. So, if DNA is the blueprint of a living organism, then replication is the process of photocopying that blueprint so every new cell exactly knows what to do. And it is the very essence of inheritance. So, biological significance and why must DNA be copied with such precision? Why it must happen. So imagine our body trying to heal a cut or grow taller without a way to duplicate your genetic instructions. Would it be possible? Maybe yes, but it won't be the same as the other part of our body. So, requirement for life. There are three main reasons for replication. First is reproduction. So, this is the passing of traits to the next generation. Second is growth, moving from a single zygote to a trillion celled adult. And lastly is the tissue repair. Our skin cells, for example, are replaced constantly. Every single one of those new cells needs a perfect copy of our DNA. So, structural logic, the beauty of DNA is that its structure actually dictates how it is copied. So, we focus on two main concepts here. One is the complementary base pairing, and then the semi-conservative nature of the process. So recall the base pairing rules. Adenine always finds thymine, and cytosine always finds guanine. Because these pairs are specific, held together by two or three hydrogen bonds, the existing strand acts as a perfect template. So if you have an A on the left, the cell knows it must put a T on the right. Next is the semi-conservative nature. This key term in your exam, semi-conservative, it means we do not print a brand new double helix from scratch. So instead, we unzip the old one and build a new partner for each half. So every new DNA molecule is actually half old and half new, and this keeps the genetic line consistent. Molecular machinery. This is the critical roles of helicase and DNA polymerase. These are the workers of this process, and in IB, you need to master two specific enzymes, and that first is the helicase, which is the zipper. So we think helicase as a pair of molecules, molecular zippers or scissors or slider. Its job is to move along the double helix and break those relatively weak hydrogen bonds. And this unwinding creates the replication fork, exposing the bases so they can be read. So, next is the DNA polymerase, which is what we call the builder. So, once the strands are open, DNA polymerase steps in. It's the builder. It grabs free floating nucleotides from the surroundings and matches them to the template. Crucially, it creates the covalent phosphodiester bonds that form the sturdy backbone of the new strand. The replication process or the timeline. So, to recap the flow, first helicase unwinds the DNA and zip. Second, the strands separate. Third, free nucleotides move into place via complementary base pairing. And finally, DNA polymerase links them together into a continuous chain. It is a fast, efficient, and incredibly accurate assembly line. So here is a quick reference table. If you can distinguish between the unzipping action of helicase and the building action of polymerase, and remember that hydrogen bonds are the ones being broken while covalent bonds are being built. You have mastered the core of the topic in DNA replication. Okay, moving on. The next is the DNA profiling. PCR and electrophoresis. This is unlocking genetic identity through molecular technology and forensic science. So, as we discuss about this, it's actually giving us the knowledge about the most powerful intersections of biology and law. This is the DNA profiling. So, we will look at how we take a microscopic biological sample and turn it into a unique genetic barcode that can solve crimes or settle family histories. So, to do this, we need to master two key laboratory techniques, and that is the PCR and then the gel electrophoresis. So, very important before we get into the how, we need to understand the what. Why is my DNA different from yours? So, let's look at the fundamentals of genetic variation. So, what is DNA profiling? While we often hear about the 99.9% similarities between human beings, DNA profiling focuses entirely on that tiny 0.1% that makes us unique. Developed by Sir Alec Jeffreys, this is not about sequencing your whole genome, that would be too slow and expensive. Instead, we look at specific checkpoints in the DNA called short tandem repeats or abbreviated as STRs. So short tandem repeats or STRs is like a stutter in the genetic code. So imagine the sequence GATA repeating over and over. So I might have 5 repeats, but you might have 12. Because we get one set of chromosomes from mom and from dad, every person has two scores of every location we test. So by testing 13 to 20 different locations, the chance of two people having the exact same scores is less than 1 in a billion. A billion. So part two is the process. As we know we are looking for these repeats, but how do we actually see them in the laboratory? We are going to see the step-by-step process of turning a sample into a profile. First is the amplification via PCR. So often a crime scene only provides a tiny trace of DNA, maybe a single drop of dried blood or just a single strand of hair. So this is not enough to see under a microscope. We use PCR or polymerase chain reaction to solve this. So think of PCR as a molecular Xerox machine or photocopying machine. It finds the specific STR regions we care about and makes millions of copies of them so we have enough material to work with. So the three steps of PCR. So PCR works in a cycle of three temperatures. First, we denature. So we heat the DNA to 95 degrees Celsius to unzip the double helix. Second, we anneal, ANNEAL. So we drop the temperature to 55 degrees Celsius so primers can find the start and end of the STRs. Then finally, we extend. At 72 degrees Celsius, an enzyme called TAQ or Taq polymerase builds the new strands. So every time we run this cycle, the amount of DNA doubles. After 30 cycles, we have over a million or a billion copies. So now we have a tube of full of DNA copies, but they are all mixed together. We need to sort them by size to see how many repeats are in each fragment. So we use gel electrophoresis for this. So DNA has a negative charge. So when we turn on an electric current, the DNA crawls through a jelly-like filter toward the positive end, and small fragments, those with few repeats, zip through the holes quickly, while big fragments get stuck and move slowly. So these sorts are DNA by size or length. Now, how do we interpreting the profile? So once sorted, we use lasers to read fluorescent tags on the DNA. This gives us a digital readout. So a series of peaks or bands. We then compare these peaks to a database. If the peaks of at all like 20 locations match a suspect, we have found the person that we are looking. Another application of this is in the forensics. So in forensics, the rule is exclusion or inclusion. So if a suspect's DNA profile is missing, even one band found at the crime scene, they are excluded. So they are innocent. So if every single band matches, the statistical probability is so high that they are included as the source of the evidence. Another example or application of it is the paternity testing. So in paternity is a logic puzzle. So a child gets 50% of their DNA from their mother and 50% from their father. So in a lab, we first cancel out the bonds the child shares with the mother. Every remaining band in the child's profile must have a matching band in the biological father's profile. So if the man being tested does not have those specific bands, he cannot be the father. You will be doing both of this simulation after the video. So DNA profiling has revolutionized justice and medicine. but it relies on the simple principles of heat, electricity, and probability. So there is the DNA profiling. Last one, let's go to the topic about DNA replication, molecular mechanisms in prokaryotes, and applications. Okay, so specifically, we will look at how prokaryotes like E. coli copy their genetic blueprint with incredible speed and accuracy. So we will start with the basic required for standard level or IB, and then we will move to specialized enzymes as we go through. Okay, so fundamental replication. Um, so this is the big picture. In the in prokaryotes, replication is not just a linear start to finish process. It is dynamic, and because prokaryotic DNA is usually circular, replication begins at a specific point called the origin of replication. So the replication fork, I already mentioned this to you before. If the DNA opens up, it forms what we call a replication bubble with two forks moving in opposite directions. So key rule to remember here, DNA is always synthesized in the 5' to 3' direction. I remember mentioning that to you before. So this is because the enzymes can only add new nucleotides to 3' and of the following chain. So this creates a high-speed assembly lines that allows the bacterium to copy its entire genome in less than an hour. Okay, so the core enzymes, they are like celebrities, but we have the best celebrities here, two best celebrities. And I think I mentioned already about the helicase and then the DNA polymerase. For the advanced mechanism, the process is a bit more complicated than just unzip and build because the two strands of DNA are anti-parallel. So to manage the complexity of the lagging strand, uh, we need a specialized team. So number one is the DNA primase. The DNA polymerase is actually a bit picky. It cannot start from a scratch. So primase, primer, has to lay down a short RNA primer first to give polymerase a place to start. Next is the polymerase 3. So this is the primary worker that adds most of the nucleotide. DNA polymerase 1, its job is to clean up. It removes those RNA primers and replaces them with actual DNA. DNA ligase, because of the lagging strand, is built in fragmental, which is what we call Okazaki fragments. Ligase acts as the molecular glue to seal the sugar phosphate backbone into one continuous strand. So you might wonder how mistakes occur. So despite the speed, the error rate is roughly 1 in a billion. So this fidelity is thanks to proofreading. So if we look at the trend of synthesis, it is incredibly consistent, as you can see. So ensuring that the daughter cells receive an exact copy of instruction for life. So what's the application of this to profiling? Why does this matter to us? Well, we've taken these biological tools out of the cell and put them into the lab to create the DNA profiles. So first we use PCR, and this is essentially DNA replication in a test tube. So instead of helicase, we use heat, that is 95 degrees Celsius, to separate the strands. We use a special heat stable enzyme called TAQ polymerase or TAC polymerase to build millions of copies of specific DNA segments, usually short tandem repeats or commonly called as the STRs. So once we have a million of copies, as I just repeating myself, you are going to put it in a gel called the gel electrophoresis, where a negative charge will migrate toward a positive electrode through a jelly-like gel. So smaller fragments move through the pores of the gel much faster than large ones, creating a unique barcode of bands. Okay, so to summarize the workflow, we sample the DNA, amplify it using PCR, separate the fragments using electrophoresis, and finally analyze the pattern. So this is how we solve crimes and determine paternity and study evolutionary relationship. So if we summarize, we move from microscopic action of helicase and polymerase to the microscopic application of forensic science. So there, for any questions, you can send me a message. Goodbye.